Hello everyone, welcome back to the Beep Sciences channel. Today we are going to discuss about the current trends in metabolic engineering and synthetic biology for sustainable agriculture. The entire project has been conceptualized by Dr. Dipankar Ghosh, currently working as Associate Professor and Principal Investigator in Microbial Engineering and Algal Biotechnology Laboratory, Department of JIS University, Kolkata. Metabolic engineering and synthetic biology are fast-growing emerging area of science, which have predominantly emphasized on human health and related issues. However, this metabolic engineering and system synthetic biology needs to develop and deliver major concern towards sustainable agriculture, which is the utmost necessity. With increase in global population, agricultural sector has been facing several burning challenges to combat malnutrition, fulfillment of current food supplies, and nutritional demand. These issues are becoming more alarming due to elevated rate of global population hike, decrease in agricultural soil fertility, which are unable to support the global food supply and demands. Therefore, a major paradigm shift has to be required in the field of agricultural section through the proper implementation of metabolic engineering and synthetic biology towards amelioration of soil fertility and to improve the crop productivities. Metabolic engineering and synthetic biology has enormous potential to engineer soil biosensor, plant engineering, value-added crop-based biomolecule generation, using and utilizing plant-based metabolic enzymes and genetic network. Based on this current global scenario, the current study or presentation demonstrates an accelerated proposal for theoretical and applicative aspects of metabolic engineering, synthetic systems biology to establish and attain sustainable agriculture in the near future. So below 3 blocks has been showing what are the different 3 aspects which will allow to attain the sustainable agriculture to combat the demand. Number 1 is biosensor that will allow to understand and comprehend the soil fertility detection. Secondly, system synthetic biology that will allow to develop value-added products or biomolecules generation. Finally, the plant genetic engineering which will allow the biomass generation in the crop under the different stress environment. So what are the theoretical and applicative aspects of metabolic engineering to allow and establish sustainable agriculture? So in this diagrammatic presentation has been clearly shown that the para-nitrophenol phosphate is the substrate and it can convert it to para-nitrophenol, which can be detected by the biosensor response by color production. Now the question comes, how this para-nitrophenol phosphate has been converted into para-nitrophenol, which will indicate the color change and being detected by the different ranges of biosensor. Here, the most driving and most interesting segment is the phosphate solubilizing microbes, where it contains acid or alkaline phosphatase. Now, this acid and alkaline phosphatase is the major biocatalyst which converts the para-nitrophenol phosphate to para-nitrophenol. So it depends on the high level of inorganic phosphate level in the soil. Now, if you're trying to understand the soil fertility index, now if your soil having a high inorganic phosphate level, that will allow to inhibit this phosphatase enzyme, and this conversion will be minimized. So biosensor will give you the lowest range of indication. Now, when your inorganic phosphate level has been gradually lowering down, the phosphatase enzyme will be working more and this conversion ratio will be improved, and biosensor will allow you to give the highest range of marking or the score. So this is the way how sustainable agriculture can be attained by the development of new kind of enzyme-based biosensor. Another aspect is the iodine deficiency. Iodine deficiency is a widespread micronutrient malnutrition problem. The biofortification of crops with iodine is a recent strategy to further enrich the human diet with a potentially cost-effective and well-accepted bioavailable iodine source. Now, atrimicinin is another very important value-added biomolecule which is mostly produced by the plant metabolic pathway. Now, if we could able to functionalize this plant metabolic pathway in a E. coli or Saccharomyces cerevisiae microbial platform, that will definitely going to having a higher production rate within a shorter period of time. As you know that the plant having the very highest generation time compared to the microbial regime. So this is the another way how you can able to make different plant-derived products within a microorganism by functionalizing the plant metabolic pathway. So this is an ideal example which has been published by J. Kisling Group in Nature 2006 that atrimicinin can be get produced within host of microbial regimes that will allow indirectly towards the sustainable agriculture and its establishment. This is Dr. Ghosh has also established another metabolic pathway prediction tool. He has published 2017, which shows that another important value-added biomolecule, that is anti-cancer drug, methyl jasmonate. Usually, methyl jasmonate has been produced in the plant, but the productivity is quite low. That could not able to combat the requirement as an anti-cancer drug. So there are certain algorithm and program has been developed by Dr. Ghosh's lab, which will allow to predict certain pathway which are thermodynamically stable and entropically and enthalpy-wise very favorable metabolic pathway. So this high-ranked pathway has tried to functionalize in E. coli and Saccharomyces, where it can attain higher production rate and the extraction procedure will also reduce the cost. So overall, it will allow to establish the sustainable agriculture by using systems synthetic biological approach in microbial platform. So as a take-home message in very short summary, by system synthetic biology application that will allow to understand and design the pathways, the novel pathways, and allow them to implement and functionalize within the microbial host, that will allow to improve the productivity of your value-added products and using microorganisms as a tool. Secondly, you can able to develop some in silico enzyme screening, because enzymes are the major biocatalysts that can be used as a biosensor development that will allow to determine the soil fertility. And thirdly, CRISPR-Cas9 gene editing system can be used for blocking the competing pathway that can allow to reduce the side product or building of the less amount of toxic metabolites within the plant. And overall, the plant will have more biomass, having a of the desired target biomolecules. So these are the three triangles through which we can able to establish a sustainable agriculture to reduce the malnutrition, to improve the crop productivity, and we can able to combat the overall the gross hike of population in entire globe. So these are the certain publications through which we have tried to establish how NPK fertilizer can ameliorate the performance by increasing the soil fertility to attain the sustainable agriculture, where bioengineering approach has been established. Secondly, we have tried to isolate a nitrogen-fixing newly isolate Enterobacter GG1, which can able to improve the plant growth. We have applied this technology in green chili plant capsicum. Secondly and thirdly, we have tried to establish how algal fertilizer can be able to attain a green sustainable agriculture by reducing the greenhouse gases from the environment. So these are the major three milestones we have achieved in last 2 years while we have been working in JIS University in microbial engineering and algal biotechnology lab of Dr. Dipankar Ghosh. So these are the people, those who are involved. Especially I would like to acknowledge Anuradha and Shurjo. Those are the internship projects they have done in our laboratory in my supervision and successfully done some of the work we have discussed. And these are my PhD scholars we have been involving actively to execute and maintain the dignity of the lab. Thank you very much for listening. I hope more videos will come up slowly that will attain your more knowledge and you will get more interest in research and the development of humanity. Thank you very much.